this is Michelle from Symbio Science Education Center. So today we're going to, as the title suggested already, we're going to look into oxidative phosphorylation and chemiosmosis process, which is technically the last stage in aerobic cellular respiration. So this process happens inside the mitochondria, specifically on the inner membrane of the mitochondria. It is a very important process that helps us to make energy. As we know, cells requires energy for normal cellular activity. For example, like DNA replication, like transcription translation, and all cell division and all these different processes. So yeah, without further ado, let's get started. So first thing first is just a little recap. Aerobic respiration has 4 main steps. So the first one is what we usually say a glycolysis, okay? This is a process where it happens in the cytoplasm that glucose is broken down into 2 pyruvate molecules. And then moving on, the next step will be a link reaction, which is the process that converts this 3-carbon molecule pyruvate into a 2-carbon molecule with an addition of a coenzyme called a coenzyme A to form acetyl coenzyme A. And then the third step, which is one of the most painful steps that a lot of students tend to struggle with, and that would be the Krebs cycle, okay? Or some people refer to it as the citric acid cycle or TCA cycle, same thing. And the last stage would be this stage that we're looking at right now, okay, which is the oxidative phosphorylation, chemiosmosis. So right now we're focusing using the product that we made throughout these steps, okay, and how can we actually help ADP molecule to be phosphorylated to make ATP. So all in all, respiration is all about making energy. And as we know, energy molecule, that our energy currency in the cell is adenosine triphosphate, ATP molecule. So it is produced by phosphorylating, so adding a phosphate group onto an ADP molecule to make an ATP. So this process requires some sort of energy. And right now we're looking into a specific way to make ATP, which is called chemiosmosis. It is a process of making ATP that is coupled with the movement of hydrogen ion down a electrochemical gradient. So let's have a look into what's happening in this process here then. So first thing first is, I just want to make it easier to understand right now. So I'm not going to draw into like super detailed diagram like this. So this is the outer membrane of the mitochondria, and that would be the inner membrane of the mitochondria. So normally this is highly folded to form cristae, which increases surface area to volume ratio, which increases the rate of ATP production. But of course, in this case here, because I want to demonstrate the understanding here, that's why I'm drawing it in a straight line. But just remember, it's highly folded. And then in between a space, we call this an intermembrane space, which is a very important location where hydrogen ions accumulate. Okay, that really builds up this electrochemical gradient. And so moving on top of the inner membrane, we can actually find quite a lot of these membrane protein that sits on the inner membrane. And most of these are used as an electron carrier that forms an electron transport chain. So these are different electron carrier protein that forms the electron transport chain. So the inside of the mitochondria, of course, that is referred to as the matrix that has lots of enzyme. This is the site where link reaction and Krebs cycle occurs. So moving on, there's still one more thing on the inner membrane, and okay, let's use blue. That's an enzyme called Atp Synthase. You don't have to know how to draw this in exam, but I'm just drawing this so you would understand what's happening. So this is an enzyme, okay? ATP synthase. So as the name suggests, it's an enzyme, ACE, that synthesizes ATP molecule. Sorry, and these are all electron carrier. All right. So let's have a look then. First thing first is, as we picked up from the previous stages, no matter whether it's in glycolysis or link reaction or Krebs cycle, we definitely make quite a lot of these reducing power, such as NADPH as well as FADH2. So reduce NAD and reduce FAD, okay? They coenzyme that temporarily holds electron and hydrogen ion. So the very first step is that we're going to use these products. They would be oxidized to release this electron to these electron carrier. So reduced NAD are going to get oxidized, have their electron that, okay, let me just use another color so it's easier to see. Which color should I pick? Blue, okay. So we have their electron that is released from reduced NAD. So these electrons will then move along from one electron carrier to another electron carrier to another electron carrier, with obviously some oxidation and reduction process happening. And when electrons are moving along the ETC, they're gonna lose energy after each transfer. So if you want to know more, you can kind of Google it and search it up. You would see the zigzag diagram where this energy level just keep on like a lower rate. So anyway, so the electron will finally be transferred to a final electron acceptor, which is oxygen in human, okay? So it will be transferred to oxygen, the oxygen molecule. I have trouble choosing color. So oxygen would pick up this electron, at the same time picking up some hydrogen ion that's actually released from reduced NAD earlier. Okay, so oxygen picking up the electron from the electron transport chain, picking up the hydrogen ion to form water. Wait, let me use this. Formation of water. Okay, so yeah, water hence will be a byproduct in cellular respiration. So moving on, okay, why are we, oh, sorry, by the way, this reduced NAD after releasing electron and hydrogen ion, they would turn back into the oxidized state, so which is NAD plus, and it can continue on to be reused in link reaction and Krebs cycle. Okay, anyway, so the key thing is, why do we want to move electrons along the electron transport chain anyway? So the reason to that is because as the electrons are moving down the electron transport chain, energy is going to be energy is going to be lost, and these energy are actually used to pump hydrogen ion from. So we're going to use the energy loss in the electron transport chain to right here pump hydrogen ion from a low concentration to a high concentration from the matrix to the intermembrane space, building up a very high hydrogen ion concentration in the intermembrane space. Hence, the pH in the intermembrane space is going to be slightly acidic. So after pumping these hydrogen ion, we can see that we have created a higher concentration of hydrogen ion in the intermembrane space compared to the matrix. So here we go. We actually established a concentration gradient, an electrochemical gradient for hydrogen ion. More here, less here. So hence hydrogen ion will have a tendency to move back into the matrix from high to low concentration, and it moves through this ATP synthase enzyme, okay, like a motor. So this ATP synthase is kind of like a motor. So when the hydrogen ion is like rushing through and rushing down the ATP synthase, this provides energy for the enzyme. And what this enzyme is doing is actually doing that process that we mentioned earlier right here. Okay, so we can move this and then duplicate. So yes. Allowing ADP to pick up a phosphate and combining a phosphate to make ATP, or actually maybe I can write it like this so it's easier to understand. So as hydrogen ion is flowing down to ATP synthase, this provides, this provides energy for ADP to combine with a phosphate to make ATP, our final product. Okay, so yeah, pretty much that's really about it for chemiosmosis. So once again, why is it called oxidative phosphorylation? The oxidative part comes from the oxidation of reduced NAD, and then the phosphorylation part comes through the phosphorylation of ADP molecule to form ATP. So what's chemiosmosis again? Once again, chemiosmosis is a type of ATP production. It's a reaction that produces ATP that's coupled with the flow of hydrogen ion. So yes, we purposely wanted the electron to move along the electron transport chain. So a significant amount of energy is lost so that we can pump hydrogen ion from the matrix into the intermembrane space so that we could build up a concentration gradient between the outside and the inside. So more on the outside than inside. So hydrogen ion will then flow from high to low concentration through these ATP synthase, and obviously it's flowing through a protein. Of course, that's facilitated diffusion. Hydrogen ion flowing down ATP synthase, providing energy for the phosphorylation of ADP to form ATP. This process is called chemiosmosis. So if you're answering an essay question for IB, chemiosmosis, this term usually would give you 1 mark. Okay, so yeah, if I were you, I would just write chemiosmosis shown if I remember that. So yeah, make sure you know chemiosmosis is a type of ATP production method coupled with the flow of hydrogen ion. Okay, so I hope you guys understand a little bit more about oxidative phosphorylation and chemiosmosis. Thank you for listening, and I'll see you guys next time. Bye-bye.